So without losing much time, let's dive in. And again, let me introduce two fabulous speakers that we have. Dr. Sally Hojvat. She was a former director of Department of Microbiological Devices at the FDA, where she spent for 12 years. And you can imagine, as a director of that huge department, her role was to lead and she was responsible for review and evaluation of safety and effectiveness of all IVD microbiology devices. And she, before joining FDA, she had already spent close to 2 more decades working in the industry. And currently, she retired from FDA in 2015, I think. And then she's currently now advising partners in diagnostics as well as Gates Foundation and WHO. So please welcome Dr. Sally Hojvat here. And joining Sally is Dr. Stephen Gitterman. He is a physician and also a laboratory scientist, both MD and PhD. And he is currently the Deputy Division Director of Microbiology Devices in US FDA in CDRH. And again, you can just imagine the role brings, and he leads the oversight of large FDA review division with responsibility in vitro devices for infectious diseases, as well as complete scientific assessments of diagnostic test clearance and approval. And there's 2 great words here, clearance and approval. Maybe we'll ask about teasing that apart. And also the review of safety evaluation for adverse events. And he represents FDA and participates in global scientific meetings. So please welcome both our wonderful speakers. The way we have designed this is Sally will go first for 20 to 25 minutes, followed by an intervention by Stephen. And then we will open it up to have our other panelists join in, and then it's going to be a more conversational. Sally, thank you. It's over to you now.
Okay, well, thank you very much for allowing us to discuss our experience with FDA, both working, in my case, as a manufacturer, as well as actually working as a review division director. And so I'm going to give more of a general overview because we only have 20 minutes. I'm not going to go into the details as much as you've seen with these other presentations that have gone before us. I thought it was worthwhile just explaining a little bit more about FDA itself, who the reviewers are, who you'd be working with, where to find specific advice, and at what points in your development phase you can work with FDA. FDA is quite a flexible organization. I know that most people think that it's extremely stringent. It is on the science, but it's also flexible and willing to look at new technologies and doesn't want to be an organization that shuts down anything that's new and innovative and would be of benefit to public health. So could I have that next slide, please? It's really talking about FDA's review balance, and this is actually across FDA, both from the vaccines, medicines, and also devices. And it's a question of balancing what are the regulatory requirements, the strict sort of written legislation, and science. Next, please. And we're talking about IVDs, and FDA includes in its definition, as you can see, reagents, instruments, and systems for the diagnosis of disease. And I also wanted to point out that we also regulate those pieces that are used for collection of specimens, preparation, as well from the human body. And as a regulation, this is a little bit different in terms of in terms of human subject protection, that specimens are considered to be something that's under the jurisdiction of human subject regulations. And as somebody pointed out before, we now know that up to perhaps 80% of any medical decisions are now based on using a diagnostic test result. And the other thing I wanted to point out was these are usually aid in the diagnostic. So usually that is something you would find in the intended use. And that means that other things, the clinical history, are supposed to be taken into account in making a decision. So they're aids in the diagnostic of a disease. They can also be used for monitoring therapy, for example, and for screening uses, and also the stage of the disease, but not for surveillance. So just in terms of what's been going on with COVID-19, FDA does not regulate tests that are just used for surveillance. Next, please. Next. So the FDA organization, the three main big centers on the left is a Center for Biologics, known as CBER, and the HIV diagnostics and blood screening tests are reviewed by individuals in that center. On the right is the Center for Medicine. And as you can see, very early on, actually in 1976, there was a division in FDA to divide out devices into a different center from medicine and from vaccines. And that was in 1976, and it was called the Food, Drug, and Cosmetic Act. Next, please. So we're now talking about within CDRH, or the Center for Devices. And I wanted to, in that group, devices, there are, of course, the in vitro devices, such as stents and pacemakers, but also IVDs. And in 2003, the Office for In Vitro Diagnostic was separated away from devices, the in vitro devices. So we're talking about the office now that would be working with you with your in vitro diagnostic tests. And there are four main divisions there: the Division of Microbiology, the Division of Chemistry and Toxicology, the Division of Immunology and Hematology, and the Division of Molecular Genetics and Pathology. And the majority of devices that you would be probably sending to FDA would be reviewed by the Division of Microbiology. There are some tests for more in the immunology. For example, IL-6, they would be looked at by immunology, but the majority of them would be in the Division of Microbiology, which Steve and I were associated with. Who are the people? Who are the reviewers? They come from both industry and academic experience. We found that you do need some people who have been in industry, who know how difficult it is to make some of these devices, as well as people in the academic area that have kept up with the science. Also, you notice that we have people who've been lab technologists, because in fact, they are users of these tests, and they know the pros and cons also of a good and a not so good test. So scientists, mostly PhDs or master's degrees, with the advent of more software and hardware in these diagnostics, we have some people who are experts in that area. Physicians like Steve that are in charge of looking at adverse events that might come in post-market from a clinical point of view. Statisticians that look at the data, every single data point that you generate with your device in a clinical trial will be looked at and recalculated by a statistician. And the other interesting piece with this particular office is that within each of those divisions, we have people who are experts in the quality system and also compliance. Next, please. So again, FDA's IVD mission is benefits against risk. Next. Next, please. From the benefits, ensuring that devices and systems are safe and effective. And on the risk side, next, please. That they are safe and effective and get them to market as quickly as possible. But when I say as quickly as possible, it doesn't mean cut corners. Next, please. So the basis of the IVD device review, you've seen that, safety and effectiveness. And as you can see from these definitions, they're based on valid scientific evidence. The probability that the benefits to health from use of the device outweigh any possible risks to a patient. And from an effectiveness point of view, is there reasonable assurance, again based on valid scientific evidence, that the use of the device in the target population provides clinically significant results? Next, please. This is a typical IVD device development pathway, which should be familiar to all of you. Basic research, analytical validation, clinical validation, data analysis, and then submitting to FDA as the submission, or as it's called often, a dossier in Europe for review. Where does the quality system have to sort of come in here? It's after basic research. At that point between basic research and analytical validation is where you lock down the design of your device. FDA interacts all the way through here. I think that's something that is not well known, but there is interaction with a developer throughout this whole development pathway, if needed. Next, please. As I mentioned, it's a total product lifecycle approach. So even the reviewers, the scientific reviewers looking at the pre-market reviews do have to know a little bit more about the quality system and the compliance issues, which are more of a post-marketing type of thing. And also the people who are looking at quality and compliance are also supposed to know a little bit more about the review process. So that way, it's instead of separating things out into silos, like some regulatory bodies may do, when a submission comes in, you have a team that consists of individuals who can look at design, the results of an analytical evaluation, clinical evaluation, know something about the quality systems, and also involved with any post-marketing surveillance if something goes wrong with the product before it finally is old and gets taken off the market. Next, please. So in general, FDA regulations, and again, they come under the Food, Drug, and Cosmetic Act, and they're called the Code of Federal Regulations. And they cover these three main areas: manufacturing. You've heard about quality system. For FDA, that's a different quality system than the ISO 13485. It's 21 CFR 820, and all products are supposed to be developed under a quality system. Software and hardware, many FDA guidance documents are available. And they can be found on the FDA website. You can actually Google FDA software guidances, and they will give you detailed information of what's expected to be given to FDA in a submission. And also whether or not the software is considered to be more of a sort of major or not so major influence on the results that are generated by the assay. Included in this also a cybersecurity, definitely something new. It's interesting to know that in the IDRF, the Intended Medical Device Regulation Forum, two of the different groups are being run by an FDA individual, one being the software as a medical device and the other one as a cybersecurity guide. So FDA very much involved with the international regulatory advice on cybersecurity and software. Human subject protection, as I mentioned, specimens are covered in this. You do have to have informed consent. That comes under one of the Code of Federal Regulations, Part 50, and you have to have IRB review. There are some exceptions. There is a guidance on use of leftover remnant specimens that you can use where you do not have to have informed consent. Those are specimens that have already been tested in the laboratory, probably in somebody's refrigerator, and you can use those as long as they're anonymous and you don't have to ask for more information. You do need IRB approval to use those remnant specimens, though. Ongoing efforts to harmonize the quality system regulations with the ISO regulations. I think it's been mentioned several times today. That's ongoing. There's been a lot of effort going into that. And now there are not so many differences between 21 CFR 820 and ISO 13485 or EEC. If you want to look up to see those differences and which standards, international standards, FDA has consensus with, there is a guidance document called Voluntary Consensus Standards. So you can check whether what you have in place and what you're using for standards for design of experiments has been accepted by FDA. Other standards, of course, related to what your types of tests would be involved with are the CLSI standards and UCAST. So a lot of participation now in ongoing efforts to harmonize regulations between stringent bodies. Pre and post-market compliance product issue alerts are shared between the competent regulatory authorities. So if one authority in Europe, for example, finds a particular product with problems, performance problems, they will alert the FDA and vice versa. So there is interaction between the compliance groups of stringent bodies. Next, please. So let's get into some sort of FDA acronyms, because there's a lot of that when you read through some of these guidance documents. What are the regulatory pathways for IVDs? There are several. The pre-submission is something that I'll talk about later. The 510K pre-market notification, 510K de novo, a PMA, a pre-market application. IDEs are infrequent for most IVDs. They're not for the in vitro devices. That means it's an investigational device exemption, which allows the result to be used on a patient, which of course normally in an investigational use only product, you cannot use that as part of managing a patient during a clinical trial. And then we have emergency use authorizations, and there's a pre-emergency use authorization way of looking at a product too. And of course, Steve is heavily involved at the moment with COVID-19, absolutely swamped, so he can answer a lot of questions on FDA's EUA and pre-EUAs for COVID. Lab developed tests, or LDTs, this is something that's quite controversial. In fact, the original definition of a lab developed test is a test that's been developed by a single lab, and it's being used in that hospital lab for their patients. And they have enforcement discretion. And in fact, their regulation is more under the CMS or the government group that looks after complexity labs, high complexity labs or moderate complexity labs. So FDA doesn't regulate LDTs. Next. Next, please. So I think you've heard about risk-based regulation of IVDs, and that is what FDA uses. The more knowledge you have about a particular disease or a particular virus or bacteria, it mitigates the risk. And what is the risk? So the risk is the fact that you might get false results. Everybody assumes that isn't an IVD developer that you get perfect 100% sensitivity and specificity, and we know that's not true. So false results are likely, and would they have a low likelihood of harm to a patient? if they would, then those devices are called Class 1. Class 2 is the devices that have a moderate likelihood of harm or that the risk can be mitigated. And Class 3 are high or unknown likelihood of harm with significant risk if there's a false result. And just to give you a couple of examples, in Class 1, you might find some chemistry results. There are very few microbiology devices in Class 1, perhaps some of the agars that don't have antibiotics in them. Class 2 is where you will find pretty much all of the types of tests that you are interested in, the AST and the AMRs. And they have what's called special controls, which do define exactly what is needed to be considered a Class 2. The Class 3, not so many as before in microbiology as they used to be, but most of the HIVs, the HBVs, HPV, some of the TB tests, and some of the, that's it. So there's been a lot of down classification. In other words, moving things from Class 3 down to Class 2, based on more knowledge about a disease and more tests that have been cleared or approved by the FDA. It also works the other way around. For example, the rapid tests for influenza used to be in Class 1, and they were up classified 2 years ago to Class 2. EUAs, not a lot is known at the beginning about an emergency use type of new pandemic virus such as COVID-19, but the urgent need mitigates taking some potential risks, which we'll hear about later. Next, please. Next, please. So why is classifying devices important? Well, as I mentioned, some Class 1 and all of the Class 2 devices, and here's some terminology, they are cleared by 510K review. And what you have to have here is in a 510K, you have to show equivalence to the performance of something that's already been cleared by FDA. Class 3 devices are approved by PMA review, and that's not a question of showing equivalence. You have to show a clinical equivalence or review. It's not showing equivalence to another device. The de novo 510K review is where you have devices with new technology or a new analyte that doesn't have a predicate. In other words, there isn't a 510K reviewed device already out there. So that has to go through a process of being evaluated under a classification request to down classify it from Class 3 down to Class 2. And that has to be reviewed by FDA to make sure that that down classification to a 510K level is appropriate. EUAs are authorized, not cleared or approved, or classified request by request. And what are the differences between these review paths? Well, it's obviously the amounts of device information requested by FDA, both manufacturing information as well as performance, and may involve just analytical data, inside data generated, or mostly for microbiology devices, you do need to have some external clinical validation. The review timelines, which are timelines that have been agreed upon by FDA and developers. I think it's 4 years, there's a big discussion about the timelines, how long FDA is being given to review a particular class of device, and how long the developer has to answer the questions, if there are questions to that. So it varies. The 510K review, if it was a straight review with no issues, would be 90 days or even less. And PMAs and de novo 510Ks are about double of that. User fees, they vary, of course, because there's more time involved with FDA reviewing. Just to let you know that there is a difference in the amount you have to pay. It's much less if you're a small business manufacturer, much, much less. So be aware that, in fact, it's quite cheap in a way compared with what I originally heard. It's about, for next year, it's just over $3,000 for a 510K for a small business fee. If you're not a small business, it's more like $12,000. For a de novo, it's much more because there's a longer review. Then you're talking about $27,000 for a small business. Okay, next, please. Oh, I forgot, just 1 second, if I could go back. We talked about user fees, but just about manufacturing and clinical site inspection. Those are periodic for class 1 and 2. They're mandatory for every class 3 device. But if there is any question of fraud or a cause, a problem with a post-market with a device, then you could expect a clinical site inspection. And those are done by a different group of people than the people in the divisions, although the division compliance people are the ones that alert the inspectors in the field of what they want to look for. Next, please. So what are the principles of a successful IVD submission to FDA? Well, obviously, a very clear and precise intended use, a complete device description, scientific evidence to support that intended use, and that includes, of course, data demonstrating safety and effectiveness of the device. And labeling, of course, includes the IFU, or if it's a point of care test, a simple kind of IFU, instructions for use. In the labeling, it includes limitations and warnings, as well as performance, analytical and clinical, and UDI information. And just a point about different labeling of kits. When you're doing a clinical trial, they're labeled investigational use only, not to be used for managing patients. If it's for research while you're trying to develop a device and you send it out just to look and see what's happening, they should be labeled research use only, not to be used for diagnosis. And your final labeling of your kit, once you're cleared or approved, would be labeled as an IVD. Next, please. So we're back to this again, next, next, and next. And I wanted to point out, what does FDA do to try to get safe and effective devices and systems to market as quickly as possible? Next. So again, we go back to that IVD device development pathway, and I mentioned that stars are there right from the beginning. And let's talk about pre-submission. If FDA interacts at stages of development from basic research all the way through analytical and clinical studies for all types of assays and claims, and it's never really too early to get FDA input. If you're talking about the basic research phase, it's just basically saying, this is what we've got. You may not get a lot of feedback, but at least FDA are looking at something perhaps new and can tell you, that looks like it could be possibly a de novo 510K. But then you would keep interacting with them. Next, please. So let's talk about the pre-submission process. And by the way, this is a free consulting, basically. You can and should meet and call FDA for, they have to cover themselves by saying non-binding discussions and advice before conducting analytical and clinical studies and before submitting a marketing application. And why is FDA interested in that? Well, you know, one of the worst things is that before they introduced the pre-submission process, we would have people who would spend millions of dollars doing their clinical trial, send in an application, and then we'd say, sorry, you did the wrong things. So this is really a mutual discussion where FDA is, you tell them what you're going to do, you tell them about your device, and you tell them what the studies you're going to do for analytical and the clinical studies. And the FDA will give you an advice and say, yes, that looks good, go ahead and do it. It's a chance to address new scientific and regulatory issues at the research and feasibility phase. And again, it's very important when you're developing new technologies like many of you are doing, and there is no FDA-approved similar device. And there is a guidance available on the pre-submission process. Next, please. Other sources of FDA collaboration and interaction with IVD developers. As I mentioned, numerous FDA guidances on anything you can think of, including individual analytes, and so you should make use of that. Posted, you can find the substantial equivalence determinations for 510K summaries. You can find the CLIA waiver point of cares listed and what was used to get those through the FDA. The de novo 510Ks are all listed, and you will see the summary of the information that was given by the developer and what was obviously okay with the FDA, and that they were now able to commercialize that product. So very important, the information is all there for you to see what other people have done and was acceptable for FDA. Least burdensome information requested. It used to be that you might have, oh, I think I'd like to hear, see something like that. A reviewer can't do that anymore. They can only ask you for what's relevant, what's readily available, and not something that's impossible. So that's something that all reviewers are taught, that don't ask people to do things that are totally impossible. You do have to justify perhaps why specimens are not available, or the number of specimens, fresh specimens, are not available in the US, for example. But if you can do that and show that you made an attempt, then that's something that the FDA will find acceptable. Interactive review of a submission. Again, that's once you get given a lead reviewer of your submission, you can interact with them and they will interact with you throughout that review. And there are some new things that have been coming through. It's called a fast track review, breakthrough devices, and more information from that is in one of the references that I've given you. So we can talk about that. That's really there to try and get things through on a fast track that are new and innovative. Next, please. How much information is needed really depends on the device, the classification, and the risk consequences, as we've seen before. The intended use, next slide, is really the driving force of the scientific review. You should have that available when you go in with a pre-submission, which means that you understand the disease, the condition, how you think this test will integrate patient clinical management and public health. You have to describe who's going to be tested, where they'll be tested, and when. What are the appropriate specimens for the claims you're making, timing and handling, the stability, and how those results may be used to benefit a patient and manage a patient. Next, please. Next. So CLIA complexity, this is another sort of a section of regulations, and it's talking about basically where tests can be run. How does FDA determine where and where not a particular kind of test can be run? Is it a complicated test that can only be run, well, that can't be run perhaps in a waived facility? A waived facility, for example, would be a doctor's office. Is it too complicated? Is it one that can be run in moderate complexity lab? or is it a test that can be run only in high complexity labs? A wave test can be run, of course, in any level of complexity type of facility. Next, please. Next. And I know you have these slides, so I won't go into detail, but for example, when you have to determine also, as well as FDA, the criteria of a moderate and a high complexity test. And there are 7 criteria basically there. Knowledge about the test and the disease, the training and experience of the people who will be running the test, how difficult is the reagent and material preparation, are there many steps involved, is there a calibration set that you have to go through, are there proficiency testing materials that have to be run, can you troubleshoot easily the equipment and maintain it, and how good are you at interpreting and judging this particular test? What kinds of individuals and locations are involved there? And there's a scoring there. And so your test, if it has a total score of 12 or less, is called a moderate complexity, and 13 or more is considered high complexity and can only be run in CLIA high complexity laboratories. Next.
Ali, should we wrap up in the next couple of minutes?
Yes, we'll do.
Yeah.
So let's talk about CLIA Waiver Studies. These are point of care tests. I know many of you are developing that. It is under CDRH oversight, so HIV point of care tests are reviewed for the CLIA waiver part by CDRH. And they're really studies that have to be done to determine the use of the product in a non-lab setting by untrained users. And they do include ease of sample collection and testing, as well as the robustness of the device. There's a new way of being able to get a waiver. It's a dual 510K CLIA waiver study and submission process, which you can look up. And over-the-counter home testing. So they're automatically considered CLIA waived, but there's only one available infectious disease that's been given this designation for HIV. Many are in the pipeline coming through, and potentially also for COVID-19. So next. Just a quick importance of following good clinical practice in a clinical study. Can you use foreign data? Depends. That's the kind of question you would ask in a pre-submission. And there are some guidance documents, a Q&A on good clinical practice. So you do need to follow this. And you can see, start off with a pre-submission meeting, and at the end, you have to prepare for a possible FDA audit and inspection. Next. Next. So emergency use authorization. This is what has come into place for the COVID. How does that happen? There are 3 different groups that can actually determine or ask for emergency. The one on the left is the military. If there was some emerging risk that had a biological, a chem, rad reagent, they could ask for emergency to be declared. On the right is usually something, or in the middle, it's the Department of Homeland Security, more of an emergency, for example, for something catastrophic, flooding or an earthquake. And most of them, all of them, have been mostly at the Health and Human Services because of emerging novel pathogens such as COVID. There's an emergency declared, just as there was for COVID, and FDA will issue EUA's emergency use authorization to tests that they review. There is supposed to be an end. If there's an end of an emergency, all of those tests turn into pumpkins. Basically, you have to then apply for a 510K. There's only one declaration that's been terminated, and that was for influenza H1N1. All the others, Ebola, Zika, MERS, and of course COVID-19 are still there. Okay, I think we're nearly to the end, just a couple quickly. Next one. The FDA have issued templates which basically tell you to fill in the gaps. This is how the submissions are going in. Those can be found on the FDA EUA website. They also have extensive information through questions and answers on that website. It's a standard. The review criteria, of course, is weighed mostly on analytical data. There's no time to do extensive clinical trials. Limited clinical specimens asked for. Sourcing, it depends on availability. And because the standard for issuance may be lower because of the rapid reviews, FDA has established conditions on their use in the form of information sheets, restrictions, adverse events. And of course, post-emergency, as I mentioned, developers are encouraged to submit 510K submissions. And I think the last two, the next one and the next one, are where you can find all of this information that I've been giving you, guidance documents, in particular for AST devices. These are some of the links to special controls guidances for AST devices, et cetera, et cetera. So I think I'll hand this over now to Steve, if he wants to add anything in addition that would be more relevant perhaps to AST, to antimicrobial resistance tests, and also if you could fill us in a little bit on what's going on at the FDA with COVID-19.
Maybe we could go straight into question and answer, Stephen, if that's okay.
That is perfectly fine.
Fine. Okay. May I then bring in Betsy, Trainer, and Peter as well? And hi, Betsy. Betsy with Carbex. Betsy, do you have a question that you could ask our panelists?
I do indeed. Hi, Sally. A common challenge for many companies that are developing tests for bloodstream infections is accessing patient samples prior to antibiotic treatment during the clinical studies, just due to the urgency related to treating septic patients promptly. So what are your thoughts on this topic? And do you have any guidance for companies that are working in this space?
Stephen?
Well, I can just add a little bit. One is, it is difficult. Certainly you can, but it should not, theoretically, and I will move over to something else in a second, but it should not be that difficult, because if you do have the protocol in place, as they draw the initial protocols, in most bacteremia studies or treatment, there's always blood cultures drawn. Now, in the present environment, and not to say it's going to change, most of the technologies being looked at are some type of molecular amplification technology. And to the extent antibiotics will not affect nucleic acid as immediately as they will viability and culture, it may not make as much a difference in that regard. But I certainly believe it can be done. It would be great to take this offline, but I don't see this as quite the challenge. And a lot of the companies, we have to say, have not come to us particularly with that. Having said that, one is there are a tremendous number of other challenges to do this. And again, I think in many ways we've been great advocates of clinical trial networks that in fact, will, because something like blood, again, not trying to oversimplify the problem, because we all know that banking has a lot of issues where you sample, et cetera. But still, that's something that lends itself extremely well to banking in a lot of ways. And again, for having pre-existing networks. But that particular issue, your question, I've not seen to be as problematic. But no doubt if we took it offline, you would convince me I'm wrong.
Thank you.
He wants you to send in a pre-submission.
Yeah, I can't agree with Sally more. The pre-submission, we learned so much, and to be honest, we enjoyed as well. That is a tremendous opportunity. We like to think we have a lot of very bright people who like to weigh in, and certainly a good deal of pragmatic experience, last answer notwithstanding. But I will add, and Sally had opened the door for this, right now, probably the most valuable service we provide is the pre-submission service, because even if you don't agree with us, you'll get a sense of where we're standing. And despite, Sally's last slide, which you've also put in the chat box, is invaluable. Yet, I will be frank, sometimes guidance is dated, and you know, some of the guidances you've cited, things change very quickly. To again cite Dr. Hojvat, who I not surprisingly agree with 99% of the time, the public health comes first. It's all benefit risk. And, you know, if something is important to the public health and the benefit risk, you know, sort of disagrees with our guidance, because our guidance is written a certain point in time and often is more conservative, use the pre-submission process. So I would just second what Sally did. Being from Carbex, we give you an open invitation, but because of COVID, our pre-submission process really is pretty much in hiatus for a few months because we've had to focus resources differently. But it's invaluable. It's just, if anybody gets off this call and says, I'm sending it in tomorrow, you might get an email to say your response will be delayed quite a while, and that's just a fact.
Thank you.
Thank you. Peter, you know, would you like to pose a question to both Stephen and Sally?
I would. Absolutely. I come from the perspective of a research and development scientist, not a regulatory professional. What are the key issues for assay and device developers in that design and development stage to make regulatory approval go smoothly? There's a lot of people in the audience, I think, that are in startups, academics, or entrepreneurs who maybe don't have a lot of experience. What does the development scientist need to consider in a smooth regulatory process? And maybe you could think of examples of companies that do well. What do they do? And companies that struggle, what are the hurdles and the mistakes that they make?
Stephen?
That's an excellent. Oh, Sally, please go ahead.
Yeah, sorry, Steve. Yeah, you know, that's been what I've been doing actually since I left FDA. And we used to see it at FDA too, that the jump from research and the concept of research to developing an assay is sometimes very big, as you said. It's learning about a quality system, which is not exactly what, you know, a research lab necessarily has. I know you document things, but do you document things to the point where FDA can look at them? So I think it's getting at some point into the organization or hiring a consultant who knows the quality system and knows the FDA regulatory pathways. They help those new companies through they're not hurdles, but they guide them into what's needed. So I would say that's the main thing that I would say, that you need to recognize that you need some help.
Okay, Stephen?
No, I could not agree more. I would say there's three pieces. The first piece is we always publish decision summaries. So if you have a product, the best thing to do is just take one and piece it down and see what did people do before. And looking at the most recent, obviously, would do so. So that is invaluable in giving you a sense of what was needed before. Second thing, again, is looking at the guidances. I won't memorize them, but there's some guidance. Third thing would be the pre-submission process, because this is not something any normal human could possibly decipher. Without working here, it is impossible. If people say, I'll go to the website and understand this. This is not the Washington Post. It is, you know, there's quite the art. And the last thing is exactly what Sally said. Get a consultant. It's not a process for the faint of heart. But people do it. People succeed all the time. Even, again, we've not met, but people who come in who have an idea and push it. And if it's a good idea, people will recognize it, and we recognize it as well. It can be done, but it's, you know, it's not straightforward. Unfortunately, perhaps as straightforward as it should be. But seriously, we are here to help. We want good things and novel things out there, the same way you do. So I would put those four pieces. Look at what we've done before, look at guidance, absolutely meet with us. And if you have a consultant, they will make the meeting with us and the process much smoother and help you out immeasurably. Probably the most common question we have is, who's a good consultant? And unfortunately, we can't answer that because we know who the bad ones are, but unfortunately, we can't recommend anybody in particular.
Thank you.
Thank you. We have 3 more questions, and if I can put in one more for myself, and out of those 3, one is also for Daniel. We'll go first to the first session had a question for you, Steve, which was, this is a startup that is making defibrillator, and they had asked, can you please elaborate on the current 510K submission process and timelines, if any, due to the current COVID situation? So we're talking about the EUA kind of processes. How much has it contracted the timelines in terms of the process?
Well, I will be honest, and I don't think I could talk about this in public, because if I said it slowed down, I can't say that. And I can't think it's working the way it normally does because you all know I'm a liar. But we have, we've obviously had to devote tremendous resources to COVID. I mean, there's easily over 1,000 submissions in various forms, but other processes are working. And again, things may be delayed a little bit, but I could tell you that everything else is moving forward. I would be honest, probably what has suffered most is the pre-submission process. And if anybody is involved in a 510K or has any question about a specific application, I suspect not, please feel free to call me. But I would not let COVID, I don't want to go through an old joke because not everybody might get it. But the fact is, the world has gone on despite COVID, and AMR and other issues are still very significant, as are a number of tropical diseases, etc. So our other work has not stopped. If anything has probably had to take a delay, it will be pre-submission. Does that answer your question or the question?
Yes, Anirudh, you could say yes or no on the chat box. Until then, we'll move to the next one. And there is an anonymous question that is, suppose I have an off-the-shelf hardware with all the certifications, and if I'm using my proprietary software with the hardware, do I still need to revalidate the hardware according to ASTM or ISO standards? Who wants to take this, Sally?
Well, I think there are guidance documents on just that issue of off-the-shelf software. So I would send somebody, just Google in FDA software guidances for off-the-shelf software, and you'll see at least the guidance that's out there at the moment. And by the way, as Steve said, the guidance documents do become dated, but you'll get an idea of if they change dramatically and something was going from, yes, you can do that, to no, you can't, then there would be an update. Right, Steve?
That's absolutely.
But if
I'm sorry, Sally, I keep interrupting
you. The guidance document will tell you where you would need to go with that kind of off-the-shelf software.
But that's absolutely true. And, I would say that, we're in the 21st century, although sometimes some people don't believe that. And in addition to OTS, which was, the big issue 10 years ago, there is guidance in cybersecurity. There's new guidance on EMC standards. For IVDs, interoperability, the standards on interoperability have gotten a tremendous boost through COVID. There's people probably seeing the LIVID standard and the guidance documents and recommendations about that. Just this last week, they came out. So there's been a lot of work. So one could talk about OTS at software, but things go along past there. And of course, the life cycle in software is major issues right now. It's been a tremendously aware understanding of the sophistication of software and changes. And again, I couldn't agree with Sally more. Looking at your individual circumstances and confirming with FDA that your understanding is correct, so you don't put in a submission that subsequently gets rejected on that basis. But we have a fair amount of people in our division are devoted to software reviews because it has become an expertise. We're not reviewing code, of course, but it's become quite sophisticated. But interoperability, which another of the speakers brought up, is a major issue. Very, very interesting. Sorry.
Thank you, Steve, for that. Quickly moving on, I think we'll just give a minute relief to Sally and Steve. And this is a question to Daniel. Daniel, if 30 seconds, you can say, this is a question from Priyanka, and she's saying, is there a scope that the Longitude Prize team can support a few select market-ready innovations aiming to secure regulatory approvals in the UK and EU before the final winner is announced? Do you want to take it offline, or you want to address it?
No, I'll just answer quickly. So the immediate thing to say is, for teams that are registered, we are having one-on-one support. And as Ruth said at the beginning of this meeting, it's a very, very light process. We're just going to ask for a few questions. What are your regulatory challenges? And you can have a couple of sessions with consultants that have this special expertise. Then in terms of actually helping in a more deep way, we can help through our network. We can help by trying to find funding if there's a real interest that the test has clinical utility. But I think beyond the one-on-one support with experts immediately, it will be varying degrees of support, with the exception of the Longitude winner, which the UK government also is committed to supporting to help get on the market in the UK.
Okay, thank you. Priyanka, I hope that addresses your question. And I have one question that kind of a mix of two. So the one is, I kind of referred to it before. It's the changing dynamic, new technologies are coming out. How does FDA look into new products or technology that claim to have no predicates? I mean, is there a case study like that, and what then happens? Is a question.
Okay. Yeah, Sally had a slide on that, which it's a non-issue. We will deal with it with the pre-submission process. If it is a new technology, it would be a de novo, which it's hard to appreciate from the outside, but de novo is not that much more work above a 510K in terms of the actual submission. But I would encourage, it'd be fantastic if, the more, I do not see this. Again, the person from Carbex is probably going to shoot me, but on the inside, I do not see it as that much more challenging. But the pre-submission process is tailor-made for that. So if you have a better mousetrap or a different mousetrap, and again, it's surprising how flexible sometimes we can be. You've seen some of the products we put under other regulations. It's sometimes, but the point is, I would not let that dissuade you. It's the pre-submission process is exactly for that.
Right, and there's actually a very good guidance. It's called Acceptance Review for De Novo Classification Request Draft Guidance for Industry and FDA Staff. And it takes you all the way through of how you apply with an acceptance checklist for asking for de novo classification. So that's the only sort of new bit that's there. And then you just do a regular sort of 510K. And again, as I mentioned, you can look up the decisions that have been made for de novos on FDA's website, and you have to look for a different number than for 510Ks. For 510Ks, it's like K and then 5 numbers. For the de novos, it's capital D, E, N, and then the numbers. So you can look up, for example, I'm just looking at the decision summary for the de novo request for evaluation of automatic class 3 designation for the Accelerate Pheno Test BC kit. Okay, and it tells me everything that they did to get the de novo 510K. So again, that's looking at everything is there on the FDA website to guide you through what other people have done, and also the sort of generic guidance, which most of it, by the way, some of these cover devices as well as IVDs. So you sort of have to skim through the device sections a little bit and look at what they've got down for in vitro diagnostics in the guidance documents from CDRH.
Okay, I know you're almost time up, but I just want to squeeze in this last question, which I've been thinking of asking to both of you. And this is about, you know, from a country like India, which I come from, and you did hear Malati, the first speaker, FDA provides the stewardship to a lot of regulatory agencies globally, including India. Even in COVID, if you have an FDA approval diagnostic test, you can directly take it to India. So with that, the question is, how do you think the FDA has changed over the last 20 years? I would say culturally, or the oversight, whatever you want to say. How has it changed in the last 20 years, and where do you think FDA will be by 2030, in another 10 years' time? To kickstart this, maybe Steve, and then Sally to end this.
I'll be quick, which is not unfortunately, not my forte. You know, it's hard to say. I will have to say, in my time, everything is moving to best practices. And there's an effort to, you know, their benefit risk has taken, you know, it's clearly the sense that it's the public health, far less reliance on individual opinion as much as consensus in doing the right thing. And I think that will continue. I think, and also to this, clearly the recognition and implied benefit risk is that there's a balance between safety and efficacy. I could talk about this for an hour. So let's let other people talk. But it's a long question, complicated, but also to the involvement of interoperability and the idea that everything is an ecosystem is certainly taking hold. And if I were developing a device now, perhaps not now, but within the next few years, I'd be looking into real world evidence very significantly, because that could be, it may flop out, but it could be very important. I mean, certainly there's a lot of people doing the right things. If it's possible, we will find out.
Yeah, well, I can give you an idea of, you know, having been in the industry on the other side of the table before I went to FDA, and you can't generalize, of course, but there were some sections of FDA that were very inflexible, did not seem to have the right people reviewing with the right talents, did not have anybody from industry. I think I was the first one into the office. And so the only way that I would accept taking a position at FDA was to have somebody who was the director of the office at that time, who was willing to work interactively with industry and do all of those things that I've put there: least burdensome, looking forward, being up to date with the science. And that's why I stayed for 12 years, because that did evolve over the time that I was there and continues to do and accelerate. So definitely a great improvement over the rather not very good interaction between FDA and the IVD industry that was present in the 1990s, for example. So great improvements, and as Steve said, looking forward instead of backwards and being much more open to new ideas.